everyone. Welcome to Chapter 20, Part 1. Of this video, Part 1, what we are going to do is focus mostly when it comes to anatomy and a little bit of the physiology of the heart. I will end at the cardiac action potential, where we will discuss more cardiodynamics in the second half of the lecture. So that's kind of where we're going to break this thing up. Anatomy, all the way through some of the foundational physiology up to cardiac action potential. Then we'll do the second half, will be mostly focusing on cardiodynamics, looking at things um, on that side. So, a good head and a good heart are always a formidable combination. So, you guys already have good heads, just keep those good hearts that you have too. As we jump into it, just as a reminder, if you're one of my students and you're going to be taking my exam, remember that when I set these up, I do tests on every chapter objective. Some of the things that you guys will need to be able to do are listed here, and so definitely pay some attention here, especially this list of 9 things here are things that you're always going to have to know. So as we get into this, let's really break this down and let's really begin and get a kind of an understanding about what it is, the heart, what it is, some information about it. And we're going to look at some kind of intro stuff, and then we'll focus in on some things. So what I want to first start out is just get us this idea of why we have a heart. Now, in the previous lecture, in chapter 19, we talked about that blood is this medium that carries and transports materials like nutrients and gases and hormones and things like that throughout the body. To move it, we need a heart. And the heart is this muscular pump that moves blood through miles of blood vessels. And in that it's about the size of your clenched fist, and it weighs around about on average around 306 to 388 grams. And I can tell you that that in that range would be normal. And now when my brother passed away, his was 506 grams. So very large cardiomegaly. Now it beats around about 100,000 times a day, and but in this, there's something big I want to focus on here, and that is the 3 circuits. Now, technically, here's kind of the way I'm going to look at it. Oftentimes, and you'll see me make that statement, I say technically the coronary circuit is a subdivision of the systemic circuit, but we are going to treat it as its own specific, very special circuit for a reason that becomes apparent to us. And what I want to do is really emphasize to you guys what these circuits do. Now, we have the 2 main is the pulmonary systemic, and then coronary is a subdivision of systemic. Now, each of these are going to basically tell you what it is they are bringing blood to and away from. Pulmonary, of course, is blood to and away from the lungs, and systemic circuit is to and away from the body systems. And the coronary circuit is to away from the heart muscle cells itself. So the pulmonary circuit, if you've already done lab, which you should have, we should have completed the lab by now, by this point in the semester, where we have seen the anatomy of the heart, and we discussed it, and we know the 4 chambers, and we know that blood will leave the right ventricle of the heart going out to the lungs via the pulmonary arteries. And we know that blood will enter into the lungs, and the lungs capillaries will oxygenate that blood, and the oxygenated blood returns back to the heart via the left and right pulmonary veins into the left atrium. So the circulation that has blood going from the right ventricle to the lungs back to left atrium, that is the pulmonary circuit portion. The systemic circuit portion, remember, this is dealing with body systems. So what we are dealing with body systems. So we are going to start with the much thicker left ventricle, and we'll go out to the body systems, and then we'll come back to the right atrium, which is going to be receiving blood from everything else. Remember, basically, I always say the atria are the receivers and the ventricles are the suppliers. Now, your coronary circuit, this is dealing blood completely to and from your cardiac muscle cells, the coronary arteries, cardiac veins, and that's a subdivision, but we'll talk about it blood to and from the heart muscle. Now, as we get into this, let's look anatomically, get an idea where a heart is when you guys are dealing with cardiac patients. Now, again, you're pumping through miles of vessels, but this is a hollow organ. Inside, there are hollow chambers that fill with blood, and the muscular organ squeezes to pump blood through those vessels miles throughout the body, plenty of blood vessels that we have. But where we would find the lungs is in a specific chamber called the mediastinum. Remember, the mediastinum is what you would find between the lungs. Between the left and right lungs, there's a mediastinum. Now, the mediastinum between the lungs. Now, something about the heart which is unusual is that, well, isn't entirely unusual. It's just that the fact we only have the one heart, but it's not entirely unusual that we have that kind of asymmetry to some organs, but we only have one heart. But the heart is quite asymmetrical. We talked in lab a little bit about the variety of the length in the right versus the left renal arteries and things like that, because the position of the aorta, the lengths will be different on the left and right sides of the abdominal aorta's artery branches. Well, here the heart is going to have 2/3 of its mass on the left to midline. 2/3 of the mass of the heart is left to midline. Now, if we imagine the line going right down the middle here, the sternum, right here, and if a line went down there, two-thirds of it is to the left of that. And there are two kind of anatomical landmarks of the heart. The pointed end, called the apex of the heart, the pointed end, is actually pointing downwards left. So inferiorly to the left, where the broad top, where you're going to find a lot of your great vessels attached to this area up here, is referred to as the base of the heart. The broad end is actually up more on the right side. So there is a twist to the heart. Now, of course, development plays a huge role into that, but one of the things I frequently talk about is because of this, the right ventricle, which is thinner and lower pressure, is facing forward, which thereby can be a little bit of some advantages there, not having the higher pressure left ventricle facing forward by having this as well. Um, Now, this idea, of course, knowing where this is paramount to understanding everything. So depending on what you want to do, like between third and fourth rib, certain things may be done. You guys may look at midclavicular line, third, fourth rib as areas, trying to find particular places to do procedures, etc. So now, one thing that I want to talk anatomically about with the heart, and this is extremely important that we understand, loads of clinical relevance here. So let's talk about serous membranes. Serous membranes, remember, surround organs. And in our ventral body cavity, we have a variety of serous membranes we talked about. We talked about the pericardium, the pleura, and the peritoneum. Now, the pericardium, we remember, is a serous membrane, and it surrounds the heart. Now, what I want to do is really kind of start us off with that. I'm going to move my camera back a little bit here. Just give myself a little extra room. I'm doing my lecture later in the day. I had to redo it because of some issues converting the old versions over. So let's say I have my heart here. Okay, so we have an organ, we have the heart, and this being the most anatomically correct heart you've ever seen, I know, is this here, the organ. Now, if we have a serous membrane, we should know that there will always be a body wall that will be outside of it. So we have our body wall, the outside of the thoracic cavity. And what happens is when you have this going on with an organ, there will always be multiple layers. And what we're going to do with these multiple layers is let's start off here. One that in reality, I'm going to draw it as close as I absolutely can. Let's see here. You can see why I am not a surgeon or anything like that. And so we're going to say that it is fused, and then it folds over, and then a outermore layer will be found as well. And I'm going to save some space there just so I can discuss anatomically this space and its importance. So remember, when it comes to any serous membrane, there's always two layers to it, always two of them. The one that is on the inside, the one that is actually on the organ, like the heart, is always called the visceral layer. And this is called the visceral pericardium because it is the serous membrane around the heart, the visceral pericardium. Now, also, when it comes to serous membranes, there is always going to be an outer layer that is going to be nearest to or closest to or even sometimes attached to physically the inside of the body wall. This is called the parietal layer, parietal pericardium. And there is a space between these two. And the space that we have between these two is part of the pericardial sac. We have the pericardial space here, and part of your pericardial sac, there is fluid in there. Fluid filled sac. There's a certain volume that is normal in human beings offhand. Since I no longer teach it, I honestly don't remember the exact thing. The cavity. I had space. We always call it pericardial cavity. I can just do that real quick. It's just make it a little bit nicer. And let's go with the notes. Sometimes so let's call it cavity with our notes here. I apologize. Okay. Now there is fluid in there, and we'll talk about this pretty clinically significant in conditions like congestive heart failure, things like that, infections, for example, Streptococcus pyogenes, the strep throat bacteria can come in and infect this, and you get infective pericarditis. This could cause fluid to accumulate in the pericardial cavity, which will start to squeeze on the heart a little bit, congestive heart failure. And what you also may notice is there is a particular external pressure that can happen where the heart squeezes on, and when the heart tries to lub dub, when it squeezes in, it can't relax back out, and it gets strangulated in a condition called cardiac tamponade. Very common, can happen. It's a very treatable type of shock. Cardiac tamponade. What you can basically do is if it is depending on the cause, you can draw pericardial fluid out of the pericardial cavity. If blood's coming out, for example, that would be a torn, like a aortic dissection. Aorta has been torn. Need a thoracic surgeon. That would not be something you would want to. You're going to help by drawing the fluids constantly out because this could be replaced. Now, so we have our parietal layer, we have our visceral layer, and our cavity, which is a fluid-filled sac. Now, pericardium, with its two layers, parietal and visceral, go ahead and make a very conscious note in your mind because this is important when it comes to kind of understand some pathophysiology to things that come along with pericardium, is that notice that there are three layers listed for the parietal pericardium where there's only two for the visceral. Now, the parietal pericardium is far denser, far tougher, far more rigid, far less giving. Now, can it be stretched over time? Yes. Can it be stretched very quickly, acutely? No. So this will allow things like congestive heart failure to occur because it doesn't want to give up very easily. So why is that? It has a dense, irregular connective tissue layer, the fibrous connective tissue layer that composes the outermost part of it. The fibrous tissue makes it hard to want to spread that apart, so it doesn't want to give outward, so the pressure goes inward. Now, there is a word called mesothelium. Mesothelium, remember, is what we call the simple squamous found in any kind of serous membrane. And like in the lungs, for example, mesothelium of the pleura. And then the areolar tissues that are sandwiched between this very loose connective tissues here, but it's that fibrous connective tissue layer added to this that makes it very much more tough. Now, the visceral pericardium, also called epicardium because it's upon the heart, covers the surface of the heart. So basically, this is a double duty. It plays part of the role is heart wall as well as pericardial sac. Um, the mesothelium in here, as you can see, again, just sticking outwards, and the areolar, the loose connective tissues here. And between these two is fluid, and there's a normal volume of fluid that is present here when that fluid is becoming too much. Internal pressures become to put force onto the heart, a squeezing force that can prevent normal heartbeat and congestive heart failure. As we look at that, we already mentioned the epicardium, visceral pericardium is actually double duty. It is part of the pericardial sac, but it is also double duty because it's part of the heart wall. Now, right here is a great image showing the pericardium with the fibrous layer of the pericardium, the areolar tissue, and the mesothelial layer, and then mesothelium and areolar tissue found here of the epicardium. Now, the epicardium can become infected as well. Pericardium, which epicardium, part of the pericardium, pericarditis. Then we would go inwards to the myocardium, the heart muscle tissue, cardiac muscle tissues. That is where a myocardial infarction would happen. These are the tissues that would die during an infarction, during a loss of blood flow, a vascular disease resulting in death in myocardium. The endocardium, which is innermost lining, it has this is called endothelium. Now, people sometimes ask, why is it endothelium here, not mesothelium? Well, endothelium is usually used in a blood vessel, but we also got to imagine when the heart tube, the cardiac tube, and everything forms basically from a blood vessel as things happen. Um, Endothelium is an inner lining that basically lines the inside of hollow structures like blood vessels in the heart. So endothelium is where we get of that. Endocardium has got the endothelial lining and the areolar tissue. And it covers everything, basically. It covers portions of the heart valves. It covers the myocardium. It covers the tendons, the chordae tendineae. And this is where you might get endocarditis. Again, some of these infectious agents get in. Bacterial colonies can actually grow on this, where sometimes either an injury happens or it just gets in and it grows on the surface of the endocardium, referred to as a vegetation. And this growing, of course, can damage the heart. It can lead to valve issues, things like that. Many ways that these things happen is they get there because of like strep throat. They go down in through a band of connective tissue in the throat. Oftentimes, it's called a fascial plane, and it travels down the throat, go right to your heart. Be very serious. So, heart issues can result from oral and pharyngeal issues, things like bad teeth and strep throat infections that aren't taken care of. And this is why you can end up with valve disorders and heart murmurs that are caused by someone getting strep throat, rheumatic heart. Okay. Now, as we look at these things, we also know the base, that's the upper part, that's where your great vessels are actually going to be located, inferiorly the apex. Now, one of the beautiful structures, which probably best found here later on in a image, right here, you can see one of the cardiac sulci here, a couple of the sulci here, really much better seen. But the coronary sulcus is between the atrium ventricles, not very well seen on this diagram. We don't have one that shows everything that I want to have on all of it, but the base and apex were briefly talked about before. I really was mostly talking about the coronary sulcus, but the base and apex again, because it's kind of the way we do the laboratory, and I always try to mimic as much that my lectures and labs dovetail together nicely, and that's how I like to do mine. Now, something you may remember, we talked about this quite a lot in the very first lab in AMP1 was our histology section. If you guys remember the histology lab, when we talked about the epithelial tissues, connective tissues, then we talked about muscle, we talked about the three types of muscle, and one of them we talked about was cardiac muscle. And one of the things I tell my students you want to look for are the intercalated discs. We say, yeah, these are branch cells. They are striated. They have one nucleus per cell. And then when I say look for the intercalated discs. And that's what lets each one of the cardiac muscle cells be electrically and chemically connected. If you have them chemically connected, basically by mastering the chemical charges, you end up resulting in an electrical charge. The chemical composition of the cell was mastered. It is like taming a molecular storm and harnessing the power of said molecular storm to become excited, like we did with skeletal muscle when I taught you guys that physiology in AP1. Now, what we're going to see is, guys, there are gap junctions present. Remember gap junctions? I described them in AP1 like they were friendship tubes, and that you have cell number one and cell number two, and it's like you have two neighbors and they live side by side. Like, say, maybe they live in an apartment building, or if you're in Europe, parts of Europe, you might call it a flat or what have you, and you share a wall together and you knock a hole in the wall, and one friend is like, hey mate, here, let me hand you a beer, and they share beers across between the hole in the wall. And then somebody said, well, here, let me give you some chips, and the other one sends some chips his way, so they shared snacks and drinks through the hole in the wall. Well, that's kind of what's happening here. We can chemically and electrically connect using these gap junctions found inside and pass ions through them, and also using desmosomes as a type of intercellular connection. We talk about intercellular connections in AMP1, that they can hold the cells together and then chemically and electrically connect them so that we have these guys able to be in unison. And we talked about that, how they had autorhythmicity to them, autorhythmic cells, automaticity, whatever. Um, I'm having a hard time saying autorhythmicity. They're autorhythmic, I can say that. So, what we're going to see is they have some differences in their looks. So definitely important, definitely something we need to know, definitely something I want to help you guys grasp. Now, as we go through, one of the big things I want to talk about is the heart itself. Now, what I'm actually going to do, I am going to draw this out with you guys. I am going to kind of follow this as much as possible. But what we're going to do, we're going to draw out a very simplified version of the heart, where we are going to help you understand some of the basics of the heart's anatomy, how the blood flows through it, how the basic functions are. Now, one of the nice things about this muscular pump is it can be simplified, and we can kind of break it down like this. And if you can break something down in a more simple way to understand it, it's always better to understand than memorize. And that's always been something I stress in my classes. So we're just going to draw this, and then we're going to draw a box on this side and a box on this side. And that's it, some boxes. I lovingly call this the Minecraft heart because it's all blocks and boxes. Now, this box we're going to call body systems. Okay, body systems. and this box we're going to call lungs. Now, I have had people, this, you're not doing it right. You're not doing it right. It doesn't matter which side we actually put these boxes on. What matters is everything goes the right place. And in that, you will see now, one of the things I used to do a lot in my kind of one-on-one study groups that we used to do pre-COVID was I would make every change I possibly could. I'd try to flip it around on them, really mess them up, see if we could make them think their way out of it. It's like, hey, just because this is the way I always draw it, he said, if you can't take it in a new way and go through it, then you don't really understand it. But this is the best way to kind of go about it at first. Is we're going to stick to it. I'm going to be very kind of my usual kind of let's be annoying about it. Let's be very picky about it. Now we're going to need these four colors. I'm going to use blue, red, purple, and green. My favorite colors to use for drawing most of these things. And what we want to do is really just get started. I'm going to go ahead and grab my blue pen, and we're going to come to the body systems. And what we're going to do is I'm going to go over here and do a little bit of a key here. We need a little bit of a legend, a key. And I'm going to say this is it's the beauty about these pens and this little notepad I use is if I make a little typo, deoxygenated blood, I can erase it. So this blood's going to be deoxygenated blood. Deoxygenated blood, we're going to use blue to reference deoxygenated blood. Now we know that blood is red. It gets brighter red when it becomes oxygenated. Blue is when blood is blue when it is bound to carbon dioxide, carbamino, hemoglobin, carbamino compounds are present. So what we want to do is take this from the body systems, and we're going to bring it right here to this chamber. Now, first thing, we're going to want to make sure we know what the chamber is. What chamber are we in? Well, on this side, we're going to treat this like we're looking straight on. So this is the right side, and this is the left side. Anatomically, if you're looking straight at it, the left side of the screen is the right side of the body. So this is the right, R-I-G-H-T, atrium. Now, atrium, atria are plural. So if this is a right atrium, this is obviously a left atrium. Left atrium. All right. Now, with the two atria defined, let's go ahead and just get the ventricles. We have a right ventricle. Do apologize that I got an email there. Right ventricle. And the left ventricle. Okay. Now, so with the four chambers defined, we'll use that as we go along. So we have deoxygenated blood coming from the body systems to the right atrium. Now, what vessels did it use to get there? Well, I am going to abbreviate these just for simplicity, and it is the superior vena cava and the inferior vena cava. So I'm going to have SVC for superior vena cava and IVC for inferior vena cava. So the two vena cavae are coming into the right atrium. Now we know the superior vena cava is coming from the above, and the inferior vena cava is coming from the bottom into the right atrium, but we're just going to have it coming into one place. Now, there is one other place that deoxygenated blood arrives to the right atrium. And there is a opening here when you are a fetus anyway. No, sorry, not this one. Sorry about that. I mean, that would be if I was here. I'm not doing that yet. This is the foramen of coronary sinus. I about got ahead of myself here, and I'm not going to redo the lecture for that, but foramen of coronary sinus. And sometimes I'll get ahead of myself. Coronary sinus. Now, remember, the coronary sinus is this confluence of the small cardiac vein, large cardiac vein, middle cardiac vein, posterior cardiac vein, and they all unite together into a confluence called the coronary sinus. And the coronary sinus inserts itself into the right atrium. So deoxygenated blood comes from three places: the superior vena cava, inferior vena cava and the foramen of coronary sinus into the normal functioning human heart. Now, this blood then needs to go into, there's no place to go but the right ventricle. But we got to make sure that blood flow is controlled. So I have a valve, and I'm going to draw the valve like it's a little trap door here. And this little valve is on the right side between the right atrium and right ventricle. Hence, this is an atrioventricular valve. So it is called one of the names we give to it is the right atrioventricular valve. I'm going to use AV valve for short. And another name we might call this is the tricuspid. And you may be wondering, why in the world are you making the R so much bigger than the I? It's because try to be right. I want to emphasize R in tricuspid to emphasize that it is the right AV valve. Remember, try to be right. Right atrioventricular valve, AV valve. So this blood will pass through this valve and into the right ventricle. And this right ventricle also has a trap door. And I'm going to put the trap door this way now. And this is called the pulmonary semilunar valve. Pulmonary semilunar valve. Or sometimes it's called pulmonic valve. Now, the pulmonary semilunar valve, this blood has to go through the pulmonary semilunar valve. Now, pulmonary is telling me that it is destined for the lungs. So this deoxygenated blood is going here. Now, case in point, we are leaving through the pulmonary semilunars. So these are the pulmonary arteries. Pulmonary arteries. And your pulmonary arteries bring us to our lungs. Now, what happens in the lung, very important that we understand, is that the lung is taking in oxygen and giving out CO2 to the air that's around us. So given that, the capillaries of lung are going to do something to the blood. What is it going to do? Well, it's going to add oxygen to the hemoglobin. So now we've got oxygenated blood. This blood gets oxygenated. I'm going to use red for oxygenated blood. And I don't know what just happened there, but for some reason my computer went a little bit black screen there. Maybe it's because I have not been moving the thing around. Okay, so what happens here is this blood comes out, that is oxygenated blood, comes into the left atrium. Now, this is using pulmonary veins. Now, wait a minute. I know what some of you are thinking. Some of you are like, but Mr. Ledford, Professor Ledford, Dr. Ledford, Professor Ledford, you need to stop. You're trying to tell me that an artery is blue and a vein is red, but that's not how things work. Well, it is in the pulmonary circulation. Remember the way that we remember that arteries are always away from the heart, and veins go to the heart. So the pulmonary veins carry oxygenated blood, oxygen-rich blood, to the left atrium. And there are two pulmonary veins on each side, two left, two right. And as that happens, of course, we have our trapdoor here. And that trapdoor we got here actually gets many names. 1, 2, 3 names. Now, we're going to, of course, call it the left AV valve. Left AV valve. Left atrioventricular valve. But another name given to it is the bicuspid. Bicuspid. I oftentimes say you should try before you buy. We also call this the mitral valve. Now, if you are in my classes, there's something that you should be aware of by now. If there's alternate names, you need to know them. Now, here is the way I work it with my lab exams. You guys have watched this from my lab as well. Now, if you watch this from my lab and me do this in the lab, remember, if there is more than one name for one structure, you can use any of the names you prefer on the exam. I don't really care which one, as long as it's one that we have available to us. If it's not one like, if I look at it, it's like, whoa, that is an unusual name. I'm definitely, and if I've never heard of it, I'm going to assume that something's going on, because I mean, I would like to think that generally I have more knowledge than the student would have, but sometimes people might know something I don't know, but I might assume some dishonesty is happening. But you are allowed to use different names. Now, but you need to be aware of the different names, because you know that I will come in sometimes and use the alternate names. And you also probably now are aware of the fact, based on the midterm exam, that you're going to need to know different names, because I might say, what is another name for this valve? Okay. Now, this blood is going to pass through the valve into our ventricle, into our left ventricle. Now, we need another gatekeeper. We need another trap door here. Now, this one is called the aortic semilunar valve. Sometimes it's called aortic valve. And what is going to happen is this blood will pass through it and out and to the body systems. Now, most of this is traveling in the aorta, hence aortic semilunar valve, out to the body systems. Now, what is the body doing? Is the body is consuming the oxygen and giving out CO2 as a waste product, okay? So what is happening is O2 from this blood gets consumed by body's tissues. CO2 gets added to it, and then we got to get it back to the lungs again, okay? Now, there are a couple other things I want to talk about here. Since I have done the foramen coronary sinus, in the adult heart, I'm going to do a structure here I want to talk about. Very important. This structure in the adult, well, luckily, you don't ever have to worry about it if everything happened right when your heart, after you were born. It should close up properly. Sometimes it doesn't. I actually knew a gentleman who was in his 70s and this opened up. But in the after you're born, this becomes what is called a fossa ovalis. Now, when you were a fetus, this was open. It's called the foramen ovale, and it lets the blood. Now, when you are a fetus, um, it keeps doing this, and it's so annoying. There we go. It's back. Sorry. I guess because I am just sitting here talking, it's not going to come on. So, okay. There we go. And so the fossa ovalis. Now, this is in a wall called the interatrial septum, and this is a wall called the interventricular septum. Now, there's one last thing I want to do. Something that I always do when I draw this out, and I've got to make sure that I do it now. And sometimes I am bad to paint myself in a corner. You guys will learn that about me as we go along. I paint myself in a corner sometimes when I'm drawing things out. One of the big things we must know is this ventricle wall is much thicker, much stronger, more powerful. And why that is it must pump to the body systems. And we have miles to go before we sleep. Okay. The one of the things when my brother passed away, and he was 50. I'm not out of my 30s yet. And but when he passed away, his left ventricle, now a normal male should be about 0.22 centimeters. His was 2.22 centimeters thick, very thick. And that's where a lot of the mass was. It was caused by the pathological cardiac hypertrophy, which, if you guys remember, hypertrophy is when muscles get big and swole. Okay. Now, What happens here, guys, is this is much thicker and stronger, more powerful. Okay, so a very important drawing we've done together, and I will say if you can draw this repeatedly till you have it down pat, then you will excel on any of the content that we're getting ready to talk about. Now, as we said, the right atrium receives deoxygenated blood, superior and inferior vena cava, along with the foramen and coronary sinus. Now, between the two atria, there is an intraatrial septum. The foramen ovale is normally open when you're a fetus. When you are born, the pressures that are caused by the increase in cardiac output and breathing and all that good stuff will initiate the closure of the fetal shunts, the three fetal shunts. Now, it does take up about 6 months, roughly 6 weeks, 6 months, somewhere around there. I got to remember the exact date. I'm going to put a number range between 6 weeks and 6 months. That's quite a lot, but that's a big range. But after you're born, it should close up. So by the time you're up and moving and mobile, that should be closed down and be a fossa ovalis. It's just a depression that's there, what's left after it closes. Now, if it doesn't and remains open, we call that a patent foramen ovale, and this foramen ovale being open will cause the deoxygenated blood after birth to pour into the left side of the heart, which will cause a depletion of oxygen capacity of that blood, which means less oxygen gets to muscles and the body doesn't function as well, and you get tired very quickly. Um, the pectinate muscles, what they are is the atrial muscles that squeeze the atria. They're found in the anterior wall of the atrium. And the auricles are just parts of the atria that are very expandable so that when it needs to hold a little bit extra blood, they can hold a little bit extra blood, especially the right atrium, very integral at having to do that. Now, one of the things we'll talk about in just a little bit, well, actually in the next chapter, sorry, more so, is consequences of blood not moving properly. I will talk a little bit about blood pooling and we'll talk about cardiodynamic instabilities a little bit as well. Now, what's going to happen as well is your left atrium, remember, deoxygenated. It's oxygenated blood, getting the oxygen-rich, oxygenated blood from your two left and right pulmonary veins. There's two on each side. That's a consequence of development, the way that things go through and develop. Now, the right ventricle takes that deoxygenated blood, pumps it to the lungs. Now, there's an interventricular septum separating the two ventricles. Sometimes it is also not complete. An interventricular septal defect can happen in such things as tetralogy of Fallot. Sometimes I will see that on shows. They'll be talking about a child having tetralogy of Fallot, the blue baby disorder. A great movie, by the way, if you're very interested in that, if you are fascinated with this, it's a wonderful movie called Something the Lord Made, starring Mos Def and the late great Alan Rickman. A great movie about the pioneering of the surgery that helped to fix the blue baby syndrome, dealing with tetralogy of Fallot. Amazing movie, amazing film. Loved it. Dealing with Julian, gosh, what was his name? But he was an African American gentleman who was given no rights in America at that time due to segregation, and yet pioneered the surgery, but was not given credit to it till long after, way after, but an amazingly intelligent man. And I think it shows a lot the futility and ignorance of racism. Now, the trabeculae carnae, if you look at inside the walls of the ventricles, they're ridged surface. It's kind of roughened looking in there. The chordae tendineae and the papillary muscles. Now, these are very important, and I want to kind of talk about this. I'm going to make a little side jot on the side of my drawing here, as something I like to do sometimes, because it's a little mnemonic I use, and I'm just going to draw him down here, and let's kind of talk about like our AV valve. And as one of my favorite comedians would say, if at first you don't succeed, skydiving is not for you. And let's say I wanted to go skydiving. Now, that is not something I'm ever going to do in my entire life because I am absolutely terrified of heights and I'm not going to jump out of an airplane. Now, imagine the parachute, that is the AV valve, okay? And then you are the papillary muscle. And the shoot cords, that is the chordae tendineae. Okay. And what is happening is the papillary muscles, though, they are going to pull to keep the cord taut so that the parachute doesn't blow away. If they are holding on to the valve and the ventricle squeezes, the valve should close. Now, if the cords don't stay taut, the valve will blow back up into the atria, causing what is called a prolapse or a regurg. We don't want this to happen. So the papillary muscles attach to the chordae tendineae, and they keep the valves closed during ventricular systole, opposing the force of ventricular systole, keeping the valves closed, the AV valves closed during ejection, cardiac ejection. So very important is that sometimes that they're too long or too loose, they become loosened. That can cause a regurg or a prolapse. It prevents the backflow of blood, called a prolapse. Your left ventricle is thicker because it has to pump blood through the systemic circuit. It's got to go through all the body systems. Okay. Now, there is a mnemonic I want to use to help you guys remember all the valves. Now, as we go to learn the valves, I am going to use a mnemonic here. And since we have it like this, I'm going to jot it down. I'm just going to stick the valves. Let's just draw them right here. We'll just write them here. And here's a handy way. So let's say that you found me passed out on the floor somewhere. And you're like, well, I don't know what to do. I might die. So, hey, just try something. Maybe you should try, purple is what I needed, try pulling my aorta. Maybe that'll work. Who knows? Try pulling my aorta. Now, what is try pulling my aorta? The tricuspid valve. First, blood comes through the tricuspid valve. Try pulling, then the pulmonary semilunar, then the mitral, then the aortic semilunar. Try pulling my aorta. Valve sequence. Okay? That's the valve sequence. Okay? And you will see me do loads of little silly little doodles sometimes, but that is something I always do when I do that. It helps you guys learn the valves. When I was a student, I used toilet paper my A, and you can guess what the A was. So that was what I used when I was a student to first learn those and get those into my head and know the sequence of blood flow through the heart. These are things that anyone in a healthcare field need to know, and they need to understand this very well. And oftentimes it's by having this down, and then you have a kid with a defect. You can understand that when you go to PEDS. If you're a nursing student, for example, PEDS, pediatric nursing, is one of the big things you'll do, and you will deal with those issues. So the right AV valve, try to be right, tricuspid. This is between the right atrium and right ventricle, hence atrioventricular valve. Left AV valve, left atrioventricular valve, also called the bicuspid or mitral. Remember, this is between the left atrium and left ventricle. The pulmonary semilunar, you're between the right ventricle and the pulmonary arteries. Technically, the pulmonary trunk and then pulmonary arteries. And then the aortic semilunar valve is going to be between the left ventricle and the aorta. Now, it is important to learn those because there's a lot of physiology that is at play, like understanding things like afterload and things like that. Very important to understand these valves and how they relate to the structures that they are associated to. Now, one thing I want to make sure you guys understand, and I'm going to preface this with just a statement. And I always kind of use this. This will be a statement you will hear a lot in my lectures because fluids and gases in the body are a lot like college students. And this may be something that you dealt with. Back in the day, I used to use one of our chemistry professors in the science department who was extremely we wouldn't say famous, infamous, for being extraordinarily tough and failing a lot of students. Dr. Cleveland was known to be very rough. I actually saw him a couple weeks ago. He is still ever much the Dr. Perry Cleveland that I've always known, but he would I always tell, but he would look at it. He'd tell the students that he goes, "I'm the Roundup." And it is his job to fail them, to keep them out of getting into med school if they didn't belong there. And I don't have that thing. I want to build you up. That's my job. Now, but I always think of Dr. Cleveland. I said, you walk in Dr. Cleveland's classroom, and he would be like, look to your left, look to your right. None of those he will be here after the first exam. I'm the Roundup. And you'd be like, forget this. I'm leaving. I'm going to go take another professor. Maybe you would go try to take Dr. Hammond's or one of the other chemistry professors in our department. And so that would be something that would happen a lot. And so students like to move sometimes from extremely high pressure to low pressure sometimes. Well, so does blood. So blood always flows from one chamber to another when one chamber squeezes on it and the other is relaxed. So blood flows from the right atrium to the right ventricle through the right AV valve, and this happens because the right atrium squeezes and pushes it through there, and then the right AV valve will close after the ventricle fills and the right ventricle contracts and squeezes blood out your pulmonary semilunar. Now, the same thing's also happening on the left side, and these are happening roughly simultaneous, almost exactly simultaneously. There are some very minor subsets of delays here, but not a lot. But when blood gets into the left atrium to go to the left ventricle, it goes to the left AV valve. When the left ventricle squeezes, you want to shut the left AV valve and open up the aortic semilunar and squeeze the blood out. Blood should only go one way. Backflow is prevented by the valves. The AV valves and the semilunar valves prevent blood backflowing either from vessels, large arteries, into the ventricle, or AV valves of blood from the ventricles back up to the atria, referred to as regurg. These things happen like mitral valve regurg, mitral valve prolapse, these things. So very important that you know these extra names. I mean, even as a young kid, I would hear these words being used in description about somebody having a problem in let's say. So now, the superior and inferior vena cava, we should know, is what brings blood out of the body systems to the right atrium. We know the coronary sinus is laying in the coronary sulcus. This is basically the confluence of the various coronary circulation veins, coronary veins, the great cardiac vein, small cardiac vein, middle cardiac vein, posterior cardiac vein, all unite together to do that, brings it to the right atrium. We know the pulmonary trunk is what comes out of the right ventricle that branches to the pulmonary arteries, carrying deoxygenated blood to lungs. The pulmonary veins, which there are two of them per lung, they come to the left atrium on each side of the left atrium, bringing oxygenated blood to left atrium. And the aorta takes that oxygenated blood out of the left ventricle to the body systems. Remember, arteries are always away and veins are always to the heart. And the aorta has three subsections, three anatomical divisions. There's the ascending, the arch, and the descending. So the ascending, arch, and descending aorta. Now, between the ascending and descending is an arch, so to go up and then to go down, you need to arch before you can do that. The arch is in between the going up and going down, like a candy cane. Okay, think aorta is kind of like a candy cane. Okay. Now, the ascending aorta, when it branches off, I want you guys to think with off the arch of the aorta. Aorta, basically, as you come up, the ascending aorta. Now, I always like to say that there's two major divisions, the ascending and the descending with arch in between. And I technically tend to count the arch as part of the ascending aorta. as it goes to the descending aorta. So we could always kind of put the aortic arch in the middle, and I probably should just do that. And just keep but the way the lab does it, and I was trying to be kind of consistent with both lab and lecture, and it's really not a major deal, to be honest, at this point, to say which subsection is it. It's what we important to know is what is coming off of it, and the arch is the gap, the bridge between the ascending part and the descending part. Now, the aortic arch itself, it's easy as your ABCs. A is the arch, has B, the brachiocephalic trunk, C, common carotid, and S, subclavian. Now, wait a minute. This is the brachiocephalic trunk, the left common carotid, and left subclavian. Now, if you guys have me in AP1, there is a phrase you hear me say a lot in AP1 is we in anatomy, we don't use words we don't need. So why am I saying left common carotid and left subclavian artery? Well, there is a right subclavian and a right common carotid. And you're probably wondering, where do they come from? Well, great news is that it comes from the brachiocephalic trunk. The brachiocephalic trunk is sometimes called the innominate artery, which I don't like that name. Innominate means no name, but it has a name. It's called brachiocephalic trunk. Brachio, arm, cephalic, head. So it gives rise to the right subclavian and right common carotid. Right subclavian is the brachial aspect of brachiocephalic, and the right common carotid is the cephalic aspect of brachiocephalic trunk. So the right subclavian artery gives us an upper thoracic region down to the arm on the right side. And the right common carotid gets the head and neck on the right side. So the left common carotid does the head and neck on the left. Left subclavian, upper thoracic, and left arm, and left upper thoracic. All this on the left side. Now, the descending aorta branches into what we call the thoracic aorta and abdominal aorta. Now, one of the things, and I do test on them, is the vertebral levels. It begins at T5 vertebral level, and it goes down to T12 vertebral level. And right about there, we're going to pass through the diaphragm. Now, because the diaphragm can move up and down, we're not going to give an exact precise. We're just going to use the diaphragm itself, because the diaphragm goes into a relaxed state and a contracted state, and it as it pulls downwards, as it contracts and moves back up to about T12 when it relaxes. So we're going to say the abdominal aorta begins below the diaphragm. Okay. Now, with these, you can see all these kind of branches here: aortic arch with the brachiocephalic trunk, left common carotid, left subclavian, things like that. A great image of the heart. Do love the anatomical things that you could see here. The distinctive thickness in the left ventricular wall. Some of the things that you can't see very well, though, is the differences in the numbers of cusps as well as I would like. But there are some images right here that really do great jobs showing tricuspid has 3 cusps, bicuspid has 2 cusps. Now, so the left ventricle is thicker, it's more muscular. Now, they don't hold the same amount of blood, but when they contract, they move the same amount of blood, or they should. And this is going to happen. If it doesn't, blood pools. There will be veins in the body that collects blood. Now, that can happen. Blood can pool in parts of the lungs. Blood can pool in the legs, for example. Blood pooling in the legs. It collects in the veins. And one of those things that is that they don't hold the same volume, but they move the same volume, or they should. Now, when one gets something like ventricular thickening, that might mess this up, of course. Now, if you have too much force, for example, in the right ventricle, that's a problem. It causes pulmonary hypertension. So what happens, guys, is now your left ventricle pumps harder. It's thicker because it's got to go through the entire body. Very strong, very thick, because it has the miles to go against. It has to overcome the diastolic pressure that's pushing down on it of 80 millimeters of mercury. have to overcome that. You have to form greater things, and that's why we don't want a right ventricle to do that. That's why one of the things is if you have transposition of great vessels, or basically things where the pulmonary vessels are getting something from the wrong ventricles as well. You need to watch the baby for pulmonary hypertension, as that can also reduce the efficacy of lungs. And we'll talk about that a little bit in chapter 21. And then I did this slide just to go through everything and say, hey, this is what we did. Try pulling my aorta, all the capillaries and yada yadas that we saw, that we drew out. And I'm not going to read it to you. This is just really to help you guys see that, what we drew out. And that kind of that all coincides with this drawing. And this drawing is one I'm going to urge you to do repeatedly. If you can draw this until you see it in your sleep and you know it, that's the thing. I think a lot of people think, well, studying is just I go over the notes. But if you don't involve yourself in it, there's not understanding. Understanding comes when you involve yourself. You put yourself into it. And then, like, to me, it's like when you see this in your sleep, that's when you're beginning to see that you actually really got it. And people's like, well, that's too much to expect. It's like, well, this is exactly what I had to do. And but our goal is to make sure you guys had the knowledge you need to make it and be successful. Remember, it's not just enough to make an A in a class. You've got to pass your entrance exams to get into your programs. You've got to persist through those programs. You've got to make it in the field. Without the knowledge we're giving you, that is very hard to do. Okay? And also, if you're a nursing student, I'm good friends with the person who's in charge of clinicals. So I will know how you do. I keep up with my students. I know what they do. All right. And now I told you that both atria pretty much contract at the same time. But there's a little bit. That's because of the SA node takes time to spread from the right to the left. Both ventricles will contract simultaneously as well, normally, unless there's some aberration. Okay. So let's talk a little bit about the coronary circulation. We're going to go into the cardiac conduction. We will end with our cardiac action potential. And so we have a big agenda today, big, hefty amount of material. Excuse me, I got a hiccup. Okay. So remember our coronary circulation, cardiac circuit. What this is to get blood to and from the heart. Our cardiac muscle cells need blood. They are constantly trying to beat, like I said, 100,000 times a day or more, that they beat. And when they beat, that takes energy. We know muscle contractions require energy to drive the power stroke, to break the cross bridges and re-energize the myosin heads, and so you can do a power stroke again. We know the sodium potassium ATPase pumps take ATP to also engage. So we know that muscle, when it works, requires a load of energy. And oxygen is needed to make loads of energy, and other nutrients are used to power that energy production. So any muscle needs blood supply. So the coronary arteries and cardiac veins are to get the nutrients and the wastes to and from heart muscle cells. Now, the coronary arteries, there are two that come off the base of the aorta. They're the very first vessels that come off the aorta itself are called the coronary arteries. There is a right one and there is a left one. And the right and left coronary arteries, as they branch off, blood will, basically what happens is, as the left ventricle squeezes and blood goes out the aorta, then it relaxes and blood flows back down the aorta, and the elastic recoil of aorta will cause blood to go into the coronary arteries, kind of like filling gutters as it rains off of your house, and then collecting it down the downspout. Okay? Now that goes and drains down in. Now the right coronary artery, it gets blood to the right atrial muscle, the right and the left ventricles muscles as well. But not only that, but it gets blood to the SA node and the AV node. So if you take out the main right coronary artery, you're dead. That's it. Your right atrium's gone. Both your ventricles can be damaged and gone. Not the entirety of, but a huge amount of even the left one, and the SA node and AV nodes, which means your heart's not going to be able to beat. So a marginal artery comes off of it, and a posterior interventricular artery comes off of it. Now, the left coronary artery comes off and it gets blood to the left atrium and left ventricle, and then the interventricular septum. So also has. Now, there is an artery come off it called the anterior interventricular artery, sometimes called left anterior descending, the so-called LAD, which is, we call it the Widowmaker. Now, I don't like calling it the Widowmaker, and there's a few reasons why I don't like calling it Widowmaker. Number one, it's not like it's any more extra deadly. Um, but that's not really my most common thing that I dislike calling it the Widowmaker. My number one reason to be against calling it the Widowmaker is it gives us a false belief that the number one killer of women in America, especially, is not coronary artery disease. Heart disease is the single biggest killer, and women die of heart disease. And widows are what is left when a husband dies. And this applies that it's men who die of heart attacks and not women. And there has been a misunderstanding. And there's also a lot of other things, like, for example, a lot of times, we always think like left arm starts to hurt, and this means if the skin of the left arm, that means you're having a heart attack. Sometimes women, they don't tend to actually refer pain like that. It may be in their mandible. So referred pain is also different amongst men and women, but women die of heart disease, one of the single biggest killers of women, especially in America. In the developing world, cardiovascular disease is huge. This is why AMP2 is so important. Now, circumflex, that's the smaller vessel here. Now, as we look, that is a branch off the left coronary artery. Sorry about that. I was just looking at the right when I was saying it. I do that sometimes as I'm looking at a word, I will say another word, try to say another word by looking at a different word and say the wrong word. So one of the things I may want you guys to know is which of these are branches off which, and that is something I could always ask either in lecture or in lab. Okay. Now, I do have a little mnemonic here that I'm going to use in a minute. Now, the blood goes into the foramen of coronary sinus by to get here, we have the combination of great cardiac vein, posterior cardiac vein, middle, and small cardiac vein. Now, they are all draining and receiving blood from the same areas as some of the major arteries provide blood to. For example, the great cardiac vein is pairing with and getting blood from the same area the anterior interventricular artery supplies blood to. The posterior cardiac vein is getting blood from the same area that the circumflex artery provides blood to. The posterior cardiac vein is draining the same area that the posterior interventricular artery supplies blood to. And the small cardiac vein is receiving blood from the back of the heart, and it runs alongside your coronary artery. Now, these help us to kind of learn, because either they drain blood from the same area as an artery, which means they go together with each other. They will be found kind of combined together on the heart surface. So I kind of imagine that you're trying to build a computer, trying to build a gaming PC, and you want to get your GAMPs right, PC GAMPs right. You may make sure you get your PC GAMPs right. Whatever in the world a GAMP is, you're trying to get all the GAMPs that you can get on your PC. Things like that. When I was in college, I built my own computers and everything. I don't have time for that kind of stuff now, but I used to build PCs, and so you want to get those PC GAMPs right. Well, PC, posterior cardiac vein, goes with the circumflex artery. So anatomically, this will help you guys out in lab, because this is how I teach it in lab to help you kind of find these things together. Great cardiac vein goes with the anterior interventricular artery. Middle cardiac vein goes with the posterior interventricular artery. And the SR, small cardiac vein, goes with the right coronary artery. PC GAMPs right. Okay. It's kind of an easy way to try to remember some of these if you use some mnemonics. I like mnemonics. They help me. If they help me, they might help you. There's a lot to learn and keep in your mind if you're trying to learn the human body. And there's tricks that always help it stick for the long haul. And you want to keep it beyond the exams. There are no mental dumps. You keep it. It needs to be there forever and always. If you think I'm not kidding you, when I was actually getting my blood drawn, and the phlebotomist at my doctor's office was one of my students, and I quizzed her while she took my blood. I asked her name the vein. describe the lumen. Just stuff like that. I just kept asking her questions. I was like, see, hurt me. It hurt you worse than it hurt me. Said I didn't feel it. So he was like, trust me, if I see you, I'm gonna ask you questions. All right, guys. Now, cardiac circulation. You can see these images here. Pretty good images to show these pairings. I'm not going to sit and point them all out as we did that in lab. Now, let's talk about cardiac conduction. To do this, remember, there's automaticity. Automaticity tells me that my heart beats on its own. Now, why does the heart beat on its own, and how can it do that? Well, it's because our heart muscles are stimulated without direct neural control, at least all of them. Now, there will be muscle cells, heart cells that are under neural control, but we have the ability to contract automatically. Now, if we look at the two cells, I actually got a nice, beautiful little electron micrograph. And the green cells are the conducting cells. These are specialized heart muscle cells here that are not neurons. Now, these, I do believe some of these are neurons, but these are heart muscle cells who do not contract. What they do is they just act kind of like wires of the heart muscle. They distribute electrical impulses without really producing the movement. But the contracting cells, these kind of reddish cells here, these squeeze and contract, and they work like skeletal muscle. And if you remember myself on skeletal muscle, if you guys had me, we talked about the protein date, how myosin wanted to hold hands with actin, and we talked about how mama calcium was in the kitchen, the terminal cisternae. Now, cardiac muscle don't have terminal cisternae, but mama calcium come out, and daddy troponin tropomyosin complex was standing in between myosin and actin, wouldn't let them hold hands. So mama calcium comes out and gets a hold of daddy, moves him out of the way, exposing G-actin's active site. Myosin reaches out cross bridges, power strokes pulls him in for a kiss, and that's how it all contracted. And if you remember my protein date when we went over that, this is what was going on to help us understand that physiology. Now, they work much like that, okay? It all kind of contracts and acts a lot like skeletal muscle fibers. There are some very minor differences. Of course, no terminal cisternae, only 2 troponin subunits, those kinds of biochemical cellular differences. But they will be functionally much like that. So very important as you guys go about learning this, okay? Now, the conducting system. One of the things I want to talk about with conducting system as we go through it is how is it arranged. Now, first thing I want to do is we're going to go back to our heart again. And we're going to draw our lovely, lovely heart here and our favorite heart drawing. Now, in case you guys do not know, the reason why this is the shape of the love heart is shaped like this is the space taken up when you anatomically combine 2 hearts and put them together. It is the area that would be taken up by the combination of 2 anatomical hearts. This is why this is the shape of love. Okay, there you go. You learned something today besides some physiology. Okay, so what we want to do, and I always kind of wish I had more colors. That's always the thing I need is more colors. I have paper pen colors, but I don't have very many friction pen colors, erasable ink colors here. I ordered a big box of different colors. I love my colors. Okay, now you're like, he's weird, he's silly. I know I am. Okay, now what we want to see, guys, is our SA node. Our SA node. And then let's use green and let's talk about the AV node. AV node. I'm going to use pink here. And I'm going to come off of that. And we'll use purple. And then I'll use black. We'll use black. And I'm going to teach you guys how to save his kin. Okay, so what we want to do, let's start out. The autonomic nervous system. I want you guys to think about the ANS, the autonomic nervous system, is going to be like the conductor, the maestro of the symphony. Now, one of the things the ANS is going to do is sympathetic and parasympathetic innervation. So your maestro can speed it up, slow it down. Okay. So the ANS is going to be like our conductor, the maestro. And as he's going through, we might have like our lead celloist or something here who's really kind of helping to set some things up. Okay. Now, the SA node, what's going to happen is the ANS gets this information. One of the really cool things it does is it sends signals to the AV node. Now, as it does this, it uses a bunch of little pathways that travel from the SA node to the AV node called the internodal pathways. Internodal pathways. And as they travel, they're going to converge on the AV node. Now, the AV node is kind of like, so the SA node's the kind of one who's like, come on, let's go, let's go. The AV node is more the node that's like, no, let's stop and smell the roses. And he has like, let's wait about 100 milliseconds. Fraction of a second, just wait. And he does, and he delays it. And so that's the AV node. And then the AV node, we have this little guy here called the bundle of His, or the AV bundle, the bundle of His. And the bundle of His gives rise to these guys here called the left and right bundle branches. So there's two of them, bundle branches. Now, for example, you could have a right bundle branch block or a left bundle branch block. And then the black things here are called the like a cat, Purkinje fibers. Purkinje fibers. Okay. And so basically, it's kind of, there's a handful of ways that you really might kind of think of this. The way they kind of listen is the but I always like to think the ANS is going to be the conductor, and the conductor's setting how we are going to play the piece. And then the SA node and the AV node follows, and then we go through the bundle of His bundle branches out to Purkinje fibers. And this is distributing the electrical impulses through the heart to time it so that it beats well. See, the heart has to get timed. And kind of think about it like this. One of the ways I like to think about it, and sorry that it keeps doing this annoying thing, where it just keeps freezing up on me. It's so annoying that it keeps trying to go to sleep, and I don't know if it's come back yet. I hope it has. And let's see. Test, test, test. Okay, it is at least recording. Okay, it's back. Sorry about that. I oftentimes like to kind of think of this as like the electrical system in your car, where you have like whatever it is you may use as the ignition kind of system, like stacked coils or something like that. They don't use distributors anymore. But your timing system, and then your spark plugs, and everything goes through the plug wires down to the fire, and set everything off so that every cylinder fires in the right sequence, and everything fires in the right sequence so that you turn the pistons, and the fires, and the crankshaft turns, and power goes to the wheels, and you go. So it has to be very well timed, and just like that. Okay. Now, I always say save his kin. So SA node, AV node, bundle of his Purkinje fibers. It just leaves out the bundle branches, the left and right bundle branches. Now, the conducting system, the sinoatrial node, SA node, is up there at up at the top of the right atrium. about the size of a postage stamp. The atrioventricular AV valve is between the right atrium and right ventricle. It delays it. We have internodal pathways going from the SA node to AV node. Then the AV bundle or bundle his goes down to the Purkinje, to the bundle branches and then Purkinje fibers of the ventricles to make it fire. And I always say, hey, remember, if you have a patient and all of his family members are having heart attacks, you got now have to save his kin, SA node, AV node, bundle of his, and Purkinje fibers. The kind of another way to remember the basic conduction is just leaving out the bundle branches. Now, again, you'll see things like bundle branch blocks, aberrations to EKGs that you'll need to know. And so these are definitely things you'll have to know as you go to read and understand an EKG, what is happening to these different parts and why is it that you may have premature atrial contractions or delayed ventricular contractions, things like that. People might have to go in, and because they have something like ectopic foci in the atria that cause swirling and delays, or sometimes postponing, or even one is too close to the AV node and it fires and gets the AV node too soon and causes premature atrial contractions, which is actually going to cause the P wave to be upside down. A student of mine had an inverted his fiance had an inverted P wave, and I was like, chances are she has an ectopic foci and it's causing premature atrial contraction. Okay, let's talk about the SA node. Now, there are timings I'm going to give. Don't worry about the timings, but I will talk about them in a minute. Now, again, kind of upper back wall where the vena cava is. We call it the pacemaker node. It starts, your pacemaker node is what starts the contraction and it stimulates the AV node. Now, as I said, the ANS is going to be like the maestro, like the conductor of the symphony orchestra. And the vagus and the sympathetic fibers of cardiac plexus. Now, depending upon who's conducting that day, you may have somebody who wants to pep up the piece a little bit, and somebody may want to make it more somber, slow piece. Depends on who's conducting that day. Now, we know the composer's supposed to dictate that, and I'm taking some liberties with my idiom here, and that's fine. The vagus nerve, cranial nerve number 10, has parasympathetic fibers and the sympathetic cardiovascular plexus. Now, when the SA node comes in, and the vagus attaches to it, it's releasing ACH. And it's binding to muscarinic acetylcholine receptors. Remember, muscarinic acetylcholine receptors are parasympathetic effector cell receptors. And if you are sympathetic, it's going to release epinephrine, norepinephrine, especially norepinephrine here at the beta-1 adrenergic receptors of the SA node. Remember, if you have a beta blocker, it's a beta-1 adrenergic receptor blocker. Prevents the binding of norepinephrine to the beta-1 receptor to causing stimulation and increased heart rate. This is a very important class of drugs used. Loads of beta blockers. Most of them end in all. Deck the halls with beta blockers. All. So everybody's like, why don't you teach this class and this class and this class? It's like, I'm happy where I am. Okay. Now, remember, when cells are excited, they depolarize. And when you depolarize a cell, you can carry depolarizations like from one neuron to another neuron or a neuron to a muscle cell using something like a neurotransmitter. Well, here we are going to go from SA node to AV node by internodal pathway, or have to go from cell to cell through gap junction to gap junction to gap junction to gap junction until you get to the AV node. Now, that's actually why an ectopic foci can get to the AV node and other things can happen, and why an ablation gets done sometimes. Now, AV node, as I said, about 100 milliseconds, about there between the right atrium and right ventricle, and I kind of stuck him in there, so he's kind of like in there, and he's a delayer. And so SA node's like, gotta go, let's go, and AV node's like, wait, let's slow down. Let's slow down, smell the roses for about 100 milliseconds, and it delays it. Why does it do it? We want to make sure that we have time to get the ventricles filled with blood. We want to let blood leave the atria, get to the ventricles. So the AV node is really just saying, hey, let's let the ventricles get enough blood before we let the contraction happen. Now, to connect it to the ventricles, we need the AV bundle of His. Then we go to the left and right bundle branches, which goes out to the Purkinje fibers. And because the left and right bundle branches come off the bundle of His, both ventricles should contract simultaneously. Now, because the SA node is in the right atrium contracts a little bit before left atrium does, but both ventricles tend to contract at the same time. Now, the Purkinje fibers go directly to the heart muscle cells, stimulating them. Now, and it is just like that. SA node, AV node, bundle of His, bundle branches, Purkinje fibers. So I'm going to give times, but do not worry about these times. Don't even have to concern yourselves with these times, but they are important enough that I'm going to mention them, but I do not test on them. It's just not something. I just think that I have bigger fish to fry. Now, SA node gets activated. That's time 0. And when it does, that's like the starting pistol goes off. The stimulus spreads. It's like a race. We're going from the SA node. We're trying to get to the AV node. That takes us 50 milliseconds. Then we get to the AV node, and the AV node says, stop, hold on, wait 100 milliseconds, roughly. So now 150 milliseconds has gone by. And now, bam, you're off to the AV bundle, a bundle of His. You go down there, it branches, Purkinje fibers, 175 milliseconds. And then finally, those Purkinje fibers stimulate the myocardium. The myocardium starts to contract at 225 milliseconds now. Okay? The myocardium contracts, the ventricles. And that is that's milliseconds. Okay, that is pretty quick, guys. Okay, pretty quick. You're talking fractions of seconds here. Okay, so SA node, AV node, bundle of His, bundle branches, Purkinje fibers. Now, here is our internodal pathways. I actually drew the same number, almost the same number this time, but it's all right. I don't have to the same number. It's just that I want you to see that they do spread out to different areas. And I even sometimes have mine come back on the A, and it can, it's okay. All right. Now, this is the kind of the hardest thing we're going to talk about here. And then we'll be done on this section. That'll be just about halfway there is myocardial action potential, and very important topic it is that I want to help you guys understand. Now, what we're going to do to understand it, and let me help you understand it, is first off, if you know me, you know that I love to graph ions moving across cell membranes. That is a thing I love. And also, you know that I love drawing excitable cells. And you guys that have had me for AP1 kind of know that I love action potentials and excitable cells, and I get excited about it. Okay, so I'm going to draw these 4 cells. We're going to have phase 1, phase 2, phase 3, and phase 4. And let me just make sure that I did, in fact, use those exact same numbers and didn't do something good. Okay. And didn't do something silly that I didn't remember. Okay, and then we're going to come here and graph it, and basically our graph is going to look like Batman. We're basically going to be graphing Batman. Now, over time, we have changes in charge that happen in millivolts that happen over time due to the movements of ions. I was reading a book recently, and the book kind of calls this as Taming the Molecular Storm. And I just love it so much that I decided I had to use that. So that's what cells are doing. They're taming molecular storms. And if we can harness these charges and basically herd ions on each side of a cell membrane, we can begin to control the charge of the cell, harness it, and excite it at very specific times. So out of 4 stages, I've got heart muscle cells here. And what we're going to do at each stage is do something with it. Now, the first one is going to be something we should know very well by now. And let's kind of discuss this a little bit. We should know that we have ICF. We should know we have ECF. And we should know that cell membranes separate these 2 fluids. Now, all cells have this pump. And this pump should be a good friend to us by now, and it's one that you guys definitely need to know. I am going to tell you guys that this will be something, if you ever hear me mention it, if it's in my notes, it's going to be on the test. You need to know this pump. If there is one primary active transport protein, you know it's this one. Okay. Now, remember, when we did it, and if you had me when I was actually in class, and I would dance it to the students, and I would be doing my best impersonation of Psy from Gangnam Style, but it was 2 potassiums in, 3 sodiums out. Sodium, potassium, pump style, pump, 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 pump style. 2 potassiums in, 3 sodiums out. Sodium, potassium, pump style. Pump, pump, pump. Now remember, when we said this happens, remember I said this makes cells like salty bananas? Makes it a salty banana. I I never thought banana was that hard to spell, but I'm having a hard time spelling banana. Now, 2 potassium cations go in, 3 sodium cations go out. Now, with this, guys, this makes the ICF have a lot of potassium, but not very much sodium. And the ECF, remember, has a lot of sodium, but not very much potassium. Hence, salty banana. Now, when that happens, let me zoom in a little bit so you can just see this a little bit better. Oh, I'm hitting the. I can't actually see. It's too dark down here. There we go. It makes me feel old now. Okay, there we go. I'm going to zoom in just a little bit so you guys can see what I'm drawing here. Okay, now what's going to happen is all of this sodium, remember, is sodium out, potassium in, is separating charges and numbers of charges so that there is more positive out and less positive in. So the ICF, remember, gets slightly negative, and the ECF gets slightly positive. And we say this cell is polarized. Remember that? We say it's polarized. That is the phrase we used to describe it, was being polarized. So what is going to happen is a cell is in resting potential. It's about negative 90 millivolts. What I'm going to do is I'm going to go down here and put about negative 90 millivolts down here. So let's put about here negative 90. And we're going to draw that right here. And we'll stop it right there. And I'm going to put a nice phase 1 there. And I'm going to use purple. Phase 1 is resting potential. This is called resting potential. And this is when a heart muscle cell is not doing anything. But by taming the molecular storm and hurting ions across cell membrane and separating the sodium from the potassium, we now have separations of charges causing a negative end and a positive end and a concentration gradient that can be utilized now. Now, how is that going to get utilized, you wonder? Well, what's going to happen is we have gap junctions. Now, at this gap junction, what's going to happen? Let's say on this side of the cell, right here, let's say this was an autorhythmic cell. This maybe is a Purkinje fiber cell right here. And it depolarized. When it depolarizes, sodium is going to pass through this gap junction. And a little bit of sodium is going to start to come into the cell. Just a little bit. Now, you guys might know what's going on right here. This is a very important concept when it comes to excitable cells. And you guys may know this idea of threshold when it comes to me and excitable cells. Now, threshold in this case is right around this idea of negative 75, which is more positive than negative 90. So we're looking about negative 75, which is more positive. So what we are starting to do is this ICF, ECF. Remember, we said the ECF was positive. and we said the ICF was negative. When the sodium comes in, it starts to make it get more positive. And when this does, and it reaches that negative 75, what you're going to do is this negative 75 is going to open up what we call a fast sodium channel. Okay? They're fast. They open up very quick. And as they open, sodium is going to rush in, a lot of it. A lot of sodium. Now, here, remember, you're getting positive now. Now, this cell was called polarized, so now it's depolarized. And you may remember such wonderful hits from me as my sodium gates in the sodium in the cell. And they're like, I'm depolarized, you dang right. I'm depolarized. I can teach you. I have a positive charge. So what's going to happen, guys, when it hits negative 75, it's threshold. And remember, threshold when you're shooting a gun, because we're in America, I'm in America, so of course guns, because every American's armed. Okay, no, not all Americans are armed. Most of them are. I'm in Tennessee, so everybody around me is okay. So when we squeeze our trigger, you can squeeze it and it won't go off until you go click, bang. When it goes click, it goes bang. And if you squeeze it too gently, it won't go off. If you squeeze it really hard, it won't go off harder. You just got to squeeze it and have to go click and bang. Well, threshold, once you reach threshold, it makes it go. Remember, I said that an action potential is the inevitable consequence of depolarizing to threshold. So this guy, sodium rushes in, and it's like, it shoots up really fast. Like 2 days ago, the fireworks in everybody's backyard in my neighborhood, except for us, because we have dogs and we don't shoot fireworks off because our dogs hate it. So that was depolarization. We got it depolarized at threshold. Fast sodium channels let this come in. Now, what is then going to start to happen? At positive 30, and the sodium gates close. Basically, what's going to happen is you're going to kind of have these two events. When you get to that plus 30 is reached, the sodium gates close. That means no more positives coming in. But our friend, let's put him right here. 2 potassiums in. 3 sodiums out. So you're starting to lose sodium, and it's kind of like a ship is sinking. And the ship is sinking, so what we're going to try to do, so our ship is sinking, because we're losing sodium here, we're losing positive charge, and we don't want to. So now we're going to open up calcium channels. And we're going to let calcium cation come in. And what this calcium cation is going to kind of do is it's going to delay repolarization, But we also know that muscle cells contract with calcium. Remember, calcium is what comes in and moves mama out of the way and lets the active sites become exposed. So that's going to be able to call what we refer to as plateau phase. Now, let me make sure I put 2 and 3 here. Plateau phase. Now, what is going to happen is this charge is getting, again, as it gets close to 0, calcium is going to come in that delays repolarization, but also gives us the muscles for calcium to contract. Now, what's going to happen is they're called slow calcium channels, and they're going to close. And then there's slow potassium gates. Now, slow potassium gates, what they're going to do, and I always use pink for potassium, and let's just put them right here, is the slow potassium gates. Potassium is going to leave K plus, and you're starting to make the cell plummet. It's going to go down there. And then that is referred to as repolarization, because remember, it's getting negative again. So it's becoming polarized again, and it's getting negative. And all I got to do is just say, hey, guess what? Your sodium, potassium, ATPase pumps. We go back to phase one, and that gets reset. So the four phases here, and what is happening at each phase. Now, this, as I said, it looks like Batman. Okay, I always think cardiac accidental graph. It looks like Batman from the side here a little bit. So, um, it's there's a meme in there somewhere, and says our Batman graph, and that's how I see it. Okay, now, the as we do this and to understand it, it's again, we're hurting ions across cell membranes. By doing that, we are getting some charges to change, which can then, those charges can excite and unexcite heart muscle cells. To do that, to help us power the help us cause a contraction, regulate the contraction and other cell processes of this. And I know that like on MCAT and stuff, they would love to put like a graph and say what kind of action potential is this, or like a myoglobin versus hemoglobin association dissociation curve. And this is cardiac. Cardiac looks like Batman. That's just me. That's the way my brain works. My brain is a little bit weird. I know. Okay. So, as we go through this, and here I really accentuated it. I could have probably spread it out a little bit. It's fine. It's as long as we understand what's going on. I enjoyed like really kind of going through that. And then the last thing I want to do is talk about your refractory periods. Now, remember, excitable cells have periods of time when they can be excited, and they have periods of time they can't be excited. Now, when they can be excited, there is a refractory period where there's a caveat. There's a catch. Now, the absolute refractory period, that is the period of time when a cell cannot respond. Absolutely not. Absolutely cannot respond again. It is deactivated. The sodium gates, the fast sodium channels are closed and incapable of opening. Now, the relative refractory period, the sodium gates are closed but capable of opening, but under certain specific condition. There is a very short absolute refractory period, which is referred to as the time when it can respond, but it has to be relatively a greater stimulus than what was before it. Now, for example, skeletal muscles, you have to be able to stimulate them rapidly and repeatedly to maintain tension. So they have long relative refractory periods so that they can be stimulated rapidly, and short absolute refractory periods. So you need to have a long period of time that they can be rapidly stimulated, very short amount of time when they can't be stimulated again. So think skeletal muscles have to be rapidly firing to maintain tension. Cardiac muscles, they need to be able to kind of maintain a certain beat. So to keep that beat, we have short relative refractory periods, but long absolute refractory periods. There's a greater amount of time that you have to let a cardiac muscle rest before it can beat again. It has a period of time, a long period of time before it can fire again. So, this is where I'm actually ending this first lecture. I thought it was a great place. The rest of it deals mostly with cardiodynamics. So it's a great stopping point. Guys, thank you so much. I hope you enjoyed the lecture. I hope you guys find them useful. A little bit newer because somebody told me that, hey, your videos, when you do them, you're kind of boring. And I'm like, you're really nice and exciting and excited in class, but you get bored. Oh, and now I know why my computer kept going out. My computer was unplugged. So that might have done that. Sorry about that. And this is why we had those problems. So, it's kind of fun to see that. But they were saying, "Hey, you're kind of boring on your recordings. You're really excited and animated when you're in front of students." And I'm like, "I will work on that." I get in front of the students and I get excited because I'm actually helping people to understand things, and I see their faces and I see them get excited. And when you can't see it, it's just kind of like, oh, whoa. So I'm like trying to imagine you guys out there shaking your head. Yes, I understand this. I'm getting this. Yeah. So it's I'm imagining some of you guys. I'm imagining DJ and Chrissy just shaking their heads. Yes, I'm getting this. I'm understanding it. So shout out to you guys. Guys, thank you so much. I hope you find these useful. Let me know if you need anything else. And thank you so much, as always, and see you in the next one.